my gentle and of course very modern apes. Welcome to Gutsick. I'm Erica, and one of the things that we do here on this channel is we go over many of the latest and greatest finds and discoveries within the fields of paleoanthropology, primatology, biology generally, evolutionary biology. As I always say, it's mostly just stuff that I find interesting and want to share with you. And we have several papers today that I just want to briefly talk about. Now, a lot of times on this channel when I cover papers, I pick one and then we spend an hour or so talking about that paper, placing it into context, talking about the methodology, interpreting the graphs and things of that nature. But unfortunately, I do not have the time to do that for every single paper out there that I find interesting. So every now and again, I do one of these videos where we'll basically just abstract hack a lot of papers with cool new discoveries that are on my radar so that I can put them on your radar and you can read into them more in depth yourself if you like. There is a time and place for both types of videos, but today is going to be one of those rapid fire tours. On the agenda today, among other things, we have the origin of worms, mummified saber-tooth tiger cubs, and dancing gibbons. So let's get started. We've got a real Frankenstein story to start us off here. The scientists who authored this paper titled "The Emergence of Sox and POU Transcription Factors Predates the Origin of Animal Stem Cells" recreated a mouse using genes that are older than the origin of the kingdom Animalia. What does that mean? The abstract starts us off with stem cells are a hallmark of animal multicellularity, and that is absolutely true. So stem cells, as we tend to conceive of them, are like these blank cells that can be genetically told to become any type of cell in your body. That means stem cells necessarily precede multicellularity because multicellularity is typically associated with specialization. You have cells in your body that are skin cells, you have cells that are neurons, you have cells that are going to help create bone or enamel or what have you. So you need stem cells to be a multicellular animal, meaning stem cells have to show up before the evolution of multicellular animals, historically speaking. They say SOX and POU transcription factors are associated with stemness and were believed to be animal innovations, reportedly absent in their unicellular relatives. So what they're saying here is that these two types of transcription factors, SOX and POU, are associated with animals in particular to the exclusion of single cellular or unicellular relatives. This means that they're thought to be like the thing that evolved, or at least in the past have been thought to be the innovation that sort of predated and then led to the evolution of animals from these unicellular ancestors because they're going to allow for stem cells and specialization. Here we describe unicellular SOX and POU factors. So what they're saying here is actually these two types of transcription factors are not only found in animals, we have versions of them found in unicellular organisms, which means strictly speaking, there's a predecessor that existed. They weren't ex nihilo, de novo evolutionary steps. Choanoflagellate and phyllosterian SOX proteins have DNA binding specificity similar to the mammalian SOX2. Choanoflagellate, but not phyllosterian SOX, can replace SOX2 to reprogram mouse somatic cells to induced pluripotent stem cells through interacting with the mouse POU member OCT4. So historically speaking, animals have these transcription factors that are going to facilitate the development of stem cells and allow for specialization. But then it turns out, thanks to this study, that choanoflagellates, which are a really closely related group to animals, they're eukaryotes, but they're not quite members of the kingdom of Animalia, they also have this SOX transcription factor, but it's not the same type of transcription factor as the one found in animals. So this raises an interesting question, right? If you took the version that's found in choanoflagellates and you stuck it into an animal, would the animal still develop? Could it still form? Or is this version too primitive? And this is an important question to ask because if it can, in fact, induce the same type of development as the one that we find in animals, then it may be that the version found in the more primitive choanoflagellates is like an evolutionary predecessor, a transitional transcription factor. And I obviously gave you the punchline, although it was a jargon-filled punchline first, which is that choanoflagellate socks can in fact induce pluripotency in mammalian cells. So pluripotency is just the ability of stem cells to become any type of cell that you need, right? That we talked about at the beginning. And what these researchers did is they stuck the choanoflagellate socks transcription factor into mice and just waited to see if it would still develop, if the socks from choanoflagellates could do the same job as the socks2 from regular mice that's found in animals. And in figure 2 here, 2G in particular, we see the mice that developed as the result of this experiment, showing that yes, you can actually take the version of this transcription factor found in a group of organisms that aren't even animals, but are a closely related group to animals, meaning that they possess a version of this transcription factor that effectively defines the kingdom of Animalia in part, and you can take that version, that more primitive version, stick it into an animal, and the job still gets done, at least with regard to this particular transcription factor, socks replacing socks2. So you might be thinking to yourself, well, okay, so there's this version of a transcription factor found in all animals that we find in choanoflagellates, and the choanoflagellate version of this transcription factor works just fine in animals, so why don't choanoflagellates develop stem cells? Why don't they have the same multicellular specialized setup as animals do? And the answer is interesting, and it's back in the abstract. Remember, you actually have two transcription factors, not one, that are necessary to create stem cells in animals, the SOX and POU transcription factor. Choanoflagellates have the proper SOX setup to the extent that you can take the choanoflagellate SOX transcription factor and stick it into an animal, and the animal will develop as normal. But they say, in contrast, choanoflagellate POU has a distinct DNA binding profile and cannot generate stem cells. So of the two pieces to create this proper stem cell setup, choanoflagellates have one, the right working SOX transcription factor, but not both. Their POU transcription factor does not work. And if you were to stick the choanoflagellate POU transcription factor into an animal, the animal would not develop. This paper is kind of remarkable from an evolutionary perspective because what it tells us is that one of the gigantic transcription factors that is responsible for making animals for their evolution as a kingdom in the first place, that transcription factor is found in a more basic form in a group of critters that aren't even animals, in a sister group to the kingdom of Animalia. Again, to the extent of which that you can take their version and stick it into an animal and it will still work. This means one of the characteristic features of animals has a precursor. It has an earlier version of it. As always, evolution is working with what it has. It doesn't necessarily have to create something entirely new. However, the POU transcription factor may be another story. They say our findings imply that the evolution of animal stem cells might have involved the exaptation of a pre-existing set of transcription factors, where pre-animal SOX was biochemically similar to extant SOX, whilst POU factors required evolutionary innovations. So basically that one of the two pieces is basically just an evolutionary rig, while the second one may have had to emerge either from another source or it needed to undergo more drastic changes in order to work the way that it works in modern animals. And that's super cool. Every year we get closer and closer to understanding the origin of every major group, and this paper fills in another important step. Speaking of important steps, and speaking of dunking on ideas of irreducible complexity and intelligent design, which I think that this paper kind of does a good job doing, let's talk about our next paper on the list, which is titled An Ediacaran Bilaterian with an Ecdysozoan Affinity from South Australia. So you might be wondering what that means. So let's break this down piece by piece, beginning with the Ediacaran. So the Ediacaran is a chunk of time that exists immediately prior to the Cambrian. You guys have probably all heard of the Cambrian before. It's a period of time that is proposed by many to see the emergence of just a ton of different big animal and plant groups. A lot of times it's packaged in terms of this idea of the Cambrian explosion, which is often co-opted by intelligent design proponents because it's like, how could all of these phyla emerge at the same time from an evolutionary perspective. Like, there certainly must have been somebody to intervene and spark or create ex nihilo each of these major animal groups, at least. But more and more, the Cambrian explosion is being challenged from a conventional perspective, like even removing this intelligent design thread that tends to run through it these days, because we're finding precursors to these phyla that previously were thought to have just emerged in the Cambrian in this chunk of time immediately prior in the Ediacaran. This fossil that we're talking about today is an example of such a monkey wrench. So a bilaterian is an animal that is symmetric along the midline, right? So we're bilaterally symmetric. Our left and right sides mirror one another. All mammals are bilaterally symmetric. Many animals are bilaterally symmetric. The sort of opposite of bilateral symmetry is radial symmetry, which something like a starfish or a jellyfish might need, where it's like the segments are repeated, in 5 or 4 or what have you. Ecdysozoans are this group of animals that are protostomes, right, as opposed to deuterostomes, and they include a lot of pretty famous charismatic clusters of critters like arthropods, lots of worms, nematodes, things of that nature. So what this paper is saying is that they found this new fossil in South Australia of an animal that is bilaterally symmetric, whose left and right sides mirror one another, that has an affinity to ecdysozoans, so is perhaps related to the common ancestor or is the common ancestor of nematodes and arthropods and different types of worms and things like that, and that this thing can be dated to the Ediacaran earlier than we previously thought, which is really, really cool. I think it's cool in part because it means that the Cambrian explosion believers stay losing, but let's get into the abstract here, the summary. Molecular clocks in Cambrian-derived metazoans strongly suggest that a Neoproterozoic origin of many animal clades. So they're saying that when we look at the molecular clock and when we look at the fossils that we find from the Cambrian, it seems like what we're looking at is a Neoproterozoic origin for lots of animal clades, as opposed to a Cambrian origin. The Neoproterozoic as a chunk of time existed prior to the Cambrian, so this one sentence at the beginning is basically saying, more and more the Cambrian explosion seems to be an artifact of an incomplete fossil record at the time that it was proposed. The more and more that we find out, the more and more that we find there are more primitive versions of Cambrian fauna living in the Neoproterozoic in times like the Ediacaran. They say, however, fossil bilaterians are rare in the Ediacaran, and no definitive ecdysozoan body fossils are known from the Precambrian. They say, notably, the base of the Cambrian is characterized by an abundance of trace fossils attributed to priapulid worms, so those are the penis worms, suggesting that their major divisions among ecdysozoan groups occurred prior to this time. This is supported by ichnofossils, so things like footprint fossils, trace fossils really, from the latest Ediacaran or the early Cambrian, left by a plausible nematoid, although definitively attributing this inferred behavior to a crowned nematoida remains contentious in the absence of body fossils. So a lot of speculation using ichnofossils, but because there is sort of an absence of body fossils for every connection that they want to make here, it remains speculative at this time. Given the high probability of the evolution of Ecdysozoa in the Proterozoic, the otherwise prolific fossil record of the Ecdysozoa and identification of more than 100 distinct Ediacaran genera, it is striking that no Ediacaran body fossils have been confidently assigned to this group. So what they're saying is basically okay, given that all signs seem to be pointing to the origin of Ecdysozoans prior to the Cambrian, why aren't we finding any body fossils? And then they go on to say, as is classic in an abstract of a paper, no problem, because we just did do that. We just did find a body fossil from an early Ecdysozoan prior to the Cambrian. They say, here we describe Uncus zogisi from the Ediacaran member in South Australia, a smooth vermiform organism with distinct curvature and anterior-posterior differentiation. The depth relief of Uncus is unique among Ediacaran fossils and consistent with rigid outer cuticles. Ecological relationships and associated trace fossils demonstrate that Uncus was motile. That means it's capable of moving, and what they were just saying is that it has a lot of characteristics that are typically assigned to ecdysozoans. They say body morphology and the inferred style of movement are consistent with nematoida, providing strong evidence for at least an ecdysozoan affinity. This validates the Precambrian origin of Ecdysozoa, reconciling a major gap between predicted patterns of evolution evolution and the fossil record. This is a common evolutionary biology W at the expense of intelligent design advocates. I'd show you the pictures and get more into this, but this is closed access, so you can just kind of make out the wormy looking shape of this guy over here on the right. Let's move on to our next paper. Safe in the knowledge that the Cambrian explosion is an idea continues to wither away. This next paper is super cool. Probably most of you have already heard of it because it kind of made the rounds on social media. Mummy of a juvenile saber-toothed cat, Homotherium latidens, from the Upper Pleistocene of Siberia. This thing isn't very complicated. I don't feel that I need to explain a lot of it to you. They found a mummy, a frozen mummy of a baby saber-toothed cat. Here are the pictures of it. It's absolutely adorable. They've got this wonderful CT scan, it looks like here. They describe it. They show the difference of it with a lion cub, like a modern lion cub, and it's got this cute little round face. Its teeth weren't even fully erupted yet. They've got some wonderful pictures of the skull here. It's just really cool. And the implications of this are phenomenal because this little Homotherium cub, I believe they have a date on it, we'll call it roughly between 30 and 40,000 years ago because they have it based off of different calibrations here, one being 31,808 plus or minus 369, and the other being 35,471 to 37,019 years. So we'll just call it between 30 and 40,000 years of age. Obviously, what is phenomenal about this find is that we're pulling more and more frozen animals up out of the permafrost every year, and some of them are pretty old. This individual in particular is not particularly ancient, but we have found pretty old critters from the ice. And I was talking with some of my archaeology, anthropology friends about this, and I am just biding my time waiting for the day when we stumble across some poor Neanderthal or archaic Homo sapiens that's been frozen in the ice. Just imagine how much information we would be able to get from one of those guys. So I find this to be a very encouraging find, as always. One of the very few positives, I guess, of freaking global climate change is that maybe we'll find more and more of these guys. Of course, the con side is if they melt too quickly, they will rot and they will be lost to us forever. So maybe that isn't actually a positive, but just cool. It's cool. We have a frozen saber-toothed tiger cub. I don't know what'll happen. I'm sure there are people out there already saying, let's clone it and make a modern one, but I don't know how possible that is. And, just because we could doesn't mean we should, etc., etc. Our next paper is titled Audience Presence Influences Cognitive Task Performance in Chimpanzees. And this one is pretty straightforward. Actually, the next few are going to be pretty straightforward in the sense that they are primatological papers that are these guys are more like us than we previously gave them credit for, which is the trend that has been sort of capitalizing on itself for the past several decades, and yet people still like to draw the line in between humans and other animals, but I digress. This is a fun one. So, in the highlights, we see that they have 6 chimpanzees that participated in computer-generated cognitive tasks over 6 years. Their performance changed with the number and types of audience members. The audiences may influence the perception of reward, cognitive load, and concentration, which may explain why there are these differences, and that audience effects may have emerged before the development of reputation-based society. So let's read the abstract and then look at a little bit more of this paper, although I think it's, again, pretty straightforward. They say human cognitive performance can be significantly influenced by the presence of audience members. While often associated with reputation management, which is considered uniquely human, it is unclear to what degree this phenomenon is shared with non-human animals. So I don't even know if I would say that reputation management is considered uniquely human, because I think we often see a lot of unnecessary displays in other apes, unnecessary in the sense that they're sort of out of nowhere, like chest beating or, branch dragging or whatever, that's effectively trying to tell other group members that, the alpha is still strong and is still in charge. I would consider that to be reputation management, but I digress. To investigate such audience effects in chimpanzees, we recorded the performance of 6 chimps on 3 different numerical touchscreen tasks varying in difficulty and cognitive demand in the presence of variable audience member compositions over 6 years. So that's a pretty nice longitudinal study, if you will. Our results indicated that chimpanzee performance was influenced by number and types of audience present. Performance increased for most difficult tasks as the experimenter count increased, while for the easiest task, performance decreased as familiar audience and experimenter count increased. This suggests that audience effects on cognitive processing can be found in chimpanzees, and that the evolutionary roots of this trait may date back to before the development of reputation-based normative societies in humans, or it could just be that chimpanzees also have reputation-based normative societies, if you will. So I like this graphical abstract here. Audience plus easy task, performance decrease. Audience plus difficult task, performance increase. This chimp looks like something, honestly, that I would draw. I really like it. I think it's cute. You've even got like the little toes down here. So this makes sense, right? When we're performing an easy task in front of an audience, we tend to do worse because we worry that we're going to botch it and look even stupider because the task is easy. Whereas with a difficult task, we almost draw inspiration from the audience and tend to do better. I was gonna get into it more, but I'm running out of time here. I'm trying to record this before I go off to teach, so we're gonna have to move on to the next one. I'll link in the description if you want to go read up some of the details about like chimpanzee performance anxiety for yourself. The next paper is titled Neurodevelopmental Timing and Sociocognitive Development in a Prosocial Cooperatively Breeding Primate, and that is Calthrixyacus. So they're looking at marmosets here. Marmosets are kind of a classic when you're comparing prosocial behavior in humans versus other primates. And I think that's partially because they're cooperative breeders. So these guys tend to be very alloparental. So you have like a pair bonded, monogamous, if you will, group of marmosets, and then mom and dad both help take care of the baby. This looks a lot like a lot of human sort of nuclear family structures that have characterized our species for quite some time. The abstract here says primate brain development is shaped by inputs received during critical periods. These inputs differ between independent and cooperative breeders. In cooperative breeders, infants interact with multiple caregivers. We study how the neurodevelopmental timing of cooperative breeding in the common marmoset maps onto behavioral milestones. To obtain structure, function, co-constructions, we combine behavioral, neuroimaging, and neural tracing experiments. We find that brain areas critically involved in observing conspecifics interacting, 1, develop in clusters, 2, have prolonged developmental trajectories, and 3, differentiate during periods of negotiations between immatures and multiple caregivers, as well as 4, do not share stronger connectivity than with other regions. Overall, developmental timing of the social brain areas correlate with social and behavioral milestones in marmosets and, as in humans, extend into adulthood. This rich social input is likely critical for the emergence of their strong sociocognitive skills. Because humans are cooperative breeders too, these findings have strong implications for the evolution of human social cognition. Really, my favorite thing from this paper is figure 3, which shows the comparison of human milestones with the brain development here on the Y and the age here on the X for humans versus marmosets. And as you can see, they look really familiar, particularly the comparison of humans to marmosets with regard to the relative locations of the onset of puberty, the age at first reproduction, and the first and second sibling with the maximum GMV here. I just think that's neat. I don't think it's really surprising that humans and marmosets look so similar in their GMV and milestones, GMV just standing for gray matter volume, because we're both pair-bonded primates that rely a whole lot on provisioning and alloparenting. So what this suggests really is that a lot of times when this type of mating system and social system evolve in tandem with one another, even in organisms as far apart as catarrhines versus platyrrhines, they tend to do so in a similar way. They're convergent, they're homoplastic in nature, because obviously we didn't inherit this setup from a common ancestor. I think it'd be interesting to pull in another pair bonding primate to see how it functions. Gibbons are, of course, the obvious choice here, so I will reach out to the authors post-haste and let them know. Speaking of gibbons, we have a contender for best paper of the year here, at least in my book, with Dance Displays in Gibbons, Biological and Linguistic Perspectives on Structured, Intentional, and Rhythmic Body Movement. I love this. Dancing gibbons, are you kidding me? Let's read the abstract. Female crested gibbons, genus Namascus, perform, I love this, conspicuous sequences of twitching movements involving the rump and extremities. I know you, like me, were dying to see what on earth that meant. You were dying to see it in action. Fortunately, these researchers are phenomenal scientists, and they included the videos in their supplementary material. So here we have it, a member of Namascus Gabriele. There are several videos. This is just the one that I liked the best. It's the first one that I saw that I was like, yeah, that does kind of look like dancing. So let's watch our female here. Let's watch her get down. You can see her rhythmically dancing. There she is. She's moving. Wow. You know, in dark times, it's pieces of media like this that really get me up out of the bed in the morning and give me hope for tomorrow. They say, however, these dances have attracted little scientific attention and their structure and meaning remain largely obscure. I imagine that's because most people saw these dances and were like, hey, look at her, she's dancing. Isn't that great? And then these were the first people that were like, but why is she dancing? Here, we analyze close-range video recordings of captive crested gibbons, extracting descriptions of dance in 4 species of Namascus. In addition, we report results from a survey amongst relevant professionals clarifying behavioral contexts of dance in captive and wild crested gibbons. So that's actually super important. Whenever you're reading a primatological study or paper in general or observation, anytime that you can compare captive versus wild behavior, you just want to do that as much as possible, because if it's found in both, it suggests that it's much more innate, versus if it's just in captive individuals, there's always the possibility that this is sort of induced exclusively because they're captive. And if it's wild, it suggests that it's not maybe important enough to persist in alternative conditions. It's always good when you have both. They say our results demonstrate that dances in Namascus represent a common and intentional form of visual communication restricted to sexually mature females. What the heck? It's the divas only. Whilst primarily used as a proceptive signal to solicit copulation, so they're dancing to try to get some action. Dances occur in a wide range of contexts related to arousal and or frustration in captivity. A linguistically informed view of this sequential behavior demonstrates that movement within dances is organized in groups and follows an isochronous isochronous rhythm, patterns not described for visual display in other non-human primates. We argue that applying the concept of dance to gibbons allows us to expand our understanding of communication in non-human primates and develop hypotheses on the rules and regularities characterizing it. We propose that crested gibbons' dances likely evolved from less elaborate rhythmic proceptive signals similar to those found in siamangs. Although dance displays in humans and crested gibbons share a number of key characteristics, they cannot be assumed to be homologous. Nevertheless, gibbon dance represents a striking model behavior to investigate the use of complex gestural signals in hominoid primates. So the idea is that it's just like gibbons are dancing for a lot of the same reasons that humans dance, to get attention and to get busy. Okay, you guys, I just wanted to take a little bit of time and talk extremely briefly about a couple of fun papers that I saw the past 2 or 3 weeks. Obviously, I did not go super in depth into pretty much any of these papers, but that's because the ones that I want to go in depth on, I release hour-long videos where I go in depth on those. So you can expect a couple of those videos starting to roll out as we move into December. If you guys like this kind of content, if you like what I do, whether it's the dunking on pseudoscience or educational content, please consider supporting me in the free way, which is liking, commenting, and subscribing. And if you want to support me in a more financial way, you can join my Patreon. Patrons typically get early access to videos, and they almost always get their names at the ends of videos. If you want to see me talk about other things, you can follow me on Blue Sky. It's gutsickgibbon.bluesky.social. And please keep an eye out. New merch coming directly from the channel is coming soon. So I want to thank you from the bottom of my gentle and of course very modern heart for being such a curious primate, and I'll see you real soon next time.